Okay. Well, if I wasn't nervous before, this will do it.
You're doing great, Mike.
All right.
You
Good morning.
You can hear me okay, though, right?
Yeah.
Okay.
Good morning. Welcome to another Thursday edition of Medicine Grand Rounds via Zoom. Today, we're going to continue our talks on COVID-19, and joining us today will be Dr. Michael J. Holtzman. He's the Director of the Division of Pulmonary and Critical Care. He's the Selma and Herman Selden Professor of Medicine. He's also a Professor of Cell Biology and Physiology. He has a particular research interest in acute and chronic responses to respiratory viruses, and Dr. Holtzman comes to us via a long line of training, starting out first at Northwestern University School of Medicine before doing his internal medicine residency at Duke University. He then completed his fellowship in pulmonary and critical care medicine at the University of California, San Francisco, where he then spent a few years there before finally joining us here at Wash U as a professor of pulmonary and critical care medicine. He holds numerous honors and awards, most notably starting out with Alpha Omega Alpha in medical school. And without going into much detail in terms of numerous awards he's won, but has finished with a Distinguished Investigator Award from the Washington University School of Medicine that he won here in 2015. Today, he's gonna talk to us more about COVID-19 updates. And without further ado, I will turn it over to him.
Thank you, Adam. And hello to everyone out there in Zoom land. Starting off here with my disclosures. Just quickly, we received funding from the NIH, the Department of Defense, the CF Foundation, the Site McCanton Center, and most recently the Harrington Discovery Institute. My consulting is confined to the DSMB for one of the asthma operations, and I have some stock in a newly founded biotech known as New Peak Therapeutics that enters in a very minimal way into near the end of the talk. So what I'm going to try to do today, as the title suggests, is tell you something about COVID-19 that you haven't already heard on the gazillion blogs, emails, downloads, messages from the Chancellor and the previous several speakers, and especially try to concentrate on what I think hasn't been touched upon yet, and that is sort of practical treatment for this one, and in a particularly strong as I can say it, recognizing that this isn't the first, it won't be the last. And so this kind of guideline that I hope to give you is also key to treating the next one, which could be, perish the thought, even worse than the current one. So I want to start by just first giving you a picture of what we see from the pulmonary side, and also to thank the people who are taking care of this. This is a graph that represents admissions or sort of patient census actually in the ICU at BJH, starting at the top here with the COVID positives, the totals rather, the COVID positives and the patients under investigation. You can see that since we started a couple of weeks ago, we've gradually climbed up to a total of about 90 patients. 46 of them are in the ICU, 30 of those are on ventilators, and another 43 less severely ill are out on the general floors. So we've ramped up pretty good. You can see that we're seeing the same sort of plateau that's being seen regionally and nationally as a good sign, but we're not sure yet whether that trend will continue or not. So that's the group that we concentrate on, and on the right here, I've listed what I call the frontliners. So these are all the individuals, especially from pulmonary critical care, but also from anesthesia and neuro and surgery, who are responsible for taking care of the sickest of the sick patients that you see here. And I just wanted to, probably the most important thing I can do today is give a shout out to those individuals who are really in the midst of the frontline battlefield here and doing just a terrific job working together and working with the rest of the system at all levels. It's really been inspirational for me to watch. Some of the olders of us are even a little jealous, not being able to be part of the hero force, but I do want to just emphasize what a terrific job they've been doing and how we thank them in every way possible. So back to the subject at hand then, in terms of treatment. I know in my clinician hat, people love to talk about blinded clinical trials. So here's my view of the blinded clinical trial approach to COVID-19, starting with the old fable about the blind men trying to figure out what an elephant is by touching different parts of it and then concluding that that part represents the whole. So everybody has their favorite blinded part, and then the approach seems to be to take that part and throw it up against the wall in the old spaghetti manner, and then come up with, at the moment, 330 registered trials for COVID in clinical trials, not to mention the countless number of trials at various places that aren't being registered. So there's no shortage of attempts to figure out what will work in this disease. Just briefly, you've heard about most of these, I'm sure by now, the passive antibody immunization, antiviral monoclonals, and vaccines, which are sort of the first line of action here, and then no shortage of news about chloroquine, hydrochloroquine, with or without azithromycin, the antiviral remdesivir, any number of monoclonals that have been transferred over from previous ARDS, CRS, CAR-T, CRS syndromes are being tried here, and then a number of other approaches such as interferons, nitric oxide, kinase inhibitors, stem cells, if you can believe it, statins, other enzyme inhibitors, and of course, corticosteroids. So no shortage of things being tried, but basically in a manner that, from a scientific standpoint, is generally irrational. So how to make sense of all this, I think what I'm trying to do here for you here is put this into some context so you can understand what I'm about to tell you and hopefully understand how we can cure this thing. So I've divided it up into two general categories, not surprisingly. The first is to attack the virus, shown up here in blue. And there's generally two approaches there. One is antivirals. Remdesivir is an example of that, but any number of drugs have been developed to try to inhibit the viral machinery function. So essentially pick apart the viral proteins, figure out which one the virus needs, and then try and block it. In addition, as I just mentioned earlier, there's also trying to get antibodies against the virus. Both passive, and you know that protocol is being launched here prominently at Mayo Clinic and many other places to take antibodies from convalescent patients and then through plasmapheresis move those into sick patients for protection. And then, you know, there's a nationwide effort to develop a more active approach in terms of vaccine by any number of companies and the NIH. So the thing to emphasize here, though, that has a fairly specific approach. The other possibility, which is the one I'm going to harp on quite a bit here this morning, is to modify the host instead of attacking the virus. So the things that people talk about doing there, just in general terms, that we'll march through. One is enhancing the antiviral response. So there, you know, as I'll explain to you, there's an innate and adaptive immune response to viruses that ultimately leads to clearance of infection and recovery to the host. And it is conceivable that one could enhance that response as a means to treatment. And the other that I'll spend the most time on is to try to correct the disease response. So we have an immune response, and in some cases a stromal cell response to respiratory viruses and other viruses. Hopefully that response is beneficial to the host, clears the infection, gets you back to work where you belong. But sometimes that doesn't work out the way nature intended, and it's that aberrant response that people spend a fair amount of time trying to correct in the hopes of being able to cure the disease. The point I wanted to emphasize, especially here, is that these kinds of approaches are more general. So they're not necessarily targeting a specific virus, they're targeting the general type of response that we make to respiratory viruses. And that has a huge advantage in some ways. So as you know, for most vaccines, we're usually chasing the rearview mirror, right? So we're looking at last year's virus in order to hope that it protects against next year's virus. The same will be true of most of these approaches. We don't know what's going to happen with the next virus. Even if we fix these, so we get an antiviral to this particular COVID, or we get an antibody, or we get a vaccine, we don't know for sure that that's going to be effective in the next one, because these viruses mutate about 1 million times a second. So the hope is here, by having a more general strategy, we can not only get after this one, but also, since the response to virus is relatively stereotyped, we can be ready for the next one as well. And I'll try and give you an example of why we think that might work. Okay, so what are the viruses? They're listed here in terms of the common respiratory viruses, and not any coincidence that these are the same viruses that you see when you send off multiplex screening assays for respiratory samples. They include human rhinovirus, 200 strains of that, the ABC varieties, an enterovirus that overlaps with this group, known as EVD68, overlaps so closely that in most screens it's not discriminated one from the other. RSV, the favorite of the children, severe viral infections and closely linked to asthma. In fact, all of these now, through work in our lab, have been linked to asthma. Influenza virus, famous for the epidemics, paraflu, relatively milder disease. Metapneumovirus, again, fairly severe disease in children and immunocompromised hosts, but not much of an issue lately. The only DNA representative, adenovirus, and then the one that we want to talk about mostly today, coronavirus, that circulates in these 4 varieties, and then the pandemic and epidemic strains that we'll talk about shortly. So that's the group. Not to spend a lot of time on the virology, but as I say, 99% of these are RNA viruses. They share a very similar genomic structure in some cases. I've divided it up here into the negative strand varieties and the positive strand varieties. So starting with pneumoviruses such as RSV, HMPV, paramyxoviruses, notably parainfluenza in humans, and as I'll show you, mouse parainfluenza in mice, flu itself, A and B varieties, and then the positive strands, the picornaviruses and the coronaviruses, which have been responsible for these recent catastrophes in public health. They all have similar kinds of genomic products, and so a lot of the antiviral strategies go after these specific things, particularly the polymerases that make the virus and the proteases, as shown here, that cleave viral proteins in order to make the virus be able to assemble and replicate. So back to the host, which is really where I want to spend the time here. And as I mentioned, simple things being simple, there is a conventional immune response to respiratory viral infection, and for that matter, other type of infections, and that's depicted here. So if you look at initially after infection, there's replication of the virus, reaching a peak at 3 to 5 days or so after infection, and then hopefully going away as the immune response begins, starting with first an innate immune response. Involves pathogen recognition receptors, interferon production, interferon-stimulated gene expression, and a host of innate immune cells, and a phenotype that's often characterized by mucus production, inflammation, and so on. And then subsequent to that, if everything goes correctly, there's an adaptive immune response involving dendritic cells, killer cytotoxic CD8 T cells, CD4 and B cells, leading to antibody production that eventually gets rid of the virus completely, so that infectious virus is cleared, remnants persist, and the host returns back to normal. And I want to say from the get-go that this virus, as bad as it looks out on the street, almost certainly has this same pattern as all of the other respiratory viruses that we've ever studied. So this is the kind of response, this is the kind of response that you're looking at in these patients, and that accounts for the acute illness that you're seeing in the first week or two after infection. So to drill down on this a little bit further, I want to give you some examples of how this works and gives us some insight into why these patients are so sick. So to talk about just a bit here on the very initial battlefield, if you will, between virus and host, and that is the interferon signaling system that's responsible for immediate defense and clearance of the virus. In simple terms, there's a couple different types of interferons. They have interferon receptors. They form a transcriptional machinery that goes to the nucleus, allows for interferon-stimulated genes to be expressed, and the products of those genes have an antiviral function that's very effective in clearing these types of viruses, including the one that we're dealing with here. Now, the viruses know this, as you might guess. They know all about all these little pieces of our genome that are out there to help defend us against the viruses, and often they have ways of subverting that response. So it's that battle, if you will, between the virus trying to get rid of what we do here and us trying to prop it up in a significant way that determines the initial few days of what happens after infection, including for this type of virus. So how do we know that about coronaviruses? So this goes back to about 2012 or so in a collaboration between my lab and the famous North Carolina lab that spawned so much of the research on coronaviruses run by Ralph Barrick. And we showed back then using mouse-adapted SARS virus, so this is back the initial epidemic wave of research, that losing STAT1 function in a mouse model, much as it would be in humans, causes big problems with trying to defend against the virus. So you can see here the weight loss that occurs after this virus, shown here. Normal mice with sort of wild type conditions is markedly enhanced when you lose STAT1 function through a genetic targeting approach that accordingly gives increases in viral titers, but still relatively good clearance by virus around, like I mentioned, 7-10 days after infection, infectious virus is cleared. But what you'll see here by the histopathology, the kind of disease that you get in the wild type mice, which develops initially in the first couple days and eventually is cleared at the time that the virus goes away. And a STAT1 mouse instead progresses to the point where there's still significant both airway and parenchymal inflammation, remodeling, and altered physiology. So something about the virus, even though it's effectively cleared in this model, still causes progressive disease. And that's a point I'm going to harp on quite a bit in relation to what happens with COVID patients. So getting rid of it's a bad thing, making it better can be a good thing. So without getting into the structural biology underneath of that, I'll just tell you that we managed to build a better STAT1 by experimental approaches in the laboratory and show that there is a reserve in the system so that improving it can actually make things better. So as good as Mother Nature is in the interferon system, artificially you can even improve it. And you look, I could just illustrate that here, sort of a bottom line idea here. If you make transgenic mice that express this hyperactive or hyper-efficient STAT1 gene that we designated STAT1CC, you can see that those animals, in comparison to STAT1 native expressing mice, do much better in terms of survival, do much better in terms of clearing the virus. And if you look at the kind of damage and destruction that occurs around 9 days after infection, in this case with a severe influenza virus, you can see that the kinds of problems that you see under normal conditions are markedly attenuated in mice that have an improved STAT1 function. So that led us, as you might predict, to launch a campaign to find molecules that might mimic this effect, so-called interferon signal enhancers, what we designated as SMIAs compounds. And that approach is still ongoing now. It's hung up, I would say, at the level of FDA purgatory, but we still have some hope that that will eventually lead to some sort of practical approach. But I wanted to just make a couple more points about this system, and then we'll move back into the COVID clinical stuff. So first is nothing's as simple as you hope. So in addition to all the pieces that I just showed you, we also identified another piece of the puzzle, a PARP9 DTX3L complex, that can partner with the ones that have been known already, both in terms of attacking the virus itself, so it can degrade the viral protease, and in terms of moving on to the nucleus and helping with interferon-stimulated gene expression, which also helps control the virus. A sort of double whammy, if you will. So there's another layer to this system that's worth mentioning for the following sort of reason. And that is because we've come to realize that, like STAT1, if you get rid of this complex, at least experimentally, it causes a problem for not only the typical viruses that we study, but also the positive strand RNA viruses, such as EMCV, EVD68, and almost certainly corona as well. So we've made this mouse, and just to show you quite quickly, in comparison to the wild type animals, there's a significant decrease in survival if you're missing that gene. And a couple things about that are really interesting, I think, for the current problem. One is, this is a graph of male mice. The female mice don't seem to have nearly as much problem. In fact, there's pretty similar survival, almost at 100% under these conditions. We'll come back to that later, but for those of you who are dealing with this problem now, I recognize already that a male predominance is part of the phenotype. And the other thing I just want to touch on here, and then I promise to get back to COVID, we've used this gene and the SNPs that are in it to map who might be susceptible to respiratory viral infection. So you all know long before COVID that when somebody coughs in your family, some of the people get sick and some don't. And the same thing with all of the other respiratory viral conditions that we know about. Some patients end up with much more severe disease than others. And in fact, we have very little insight into why that might occur. So just to give you some idea that we have a crack in the door here, we've looked at this issue in terms of the SNPs that are occurring in the DTX3L gene. We've solved the structure for that complex, and we've placed those SNPs of importance at various points that we think are functionally important. And we've now been able to demonstrate that in terms of comparison to wild type conditions, when we overexpress DTX3L, you can see a decrease in viral protease expression, suggesting that it's being degraded by this complex, whereas some of the SNPs, although not all of them, lose that capacity. And you can see those just marching along here. So we lose the decrease, that means we lose the functional effect. So these SNPs convey some defect in being able to handle the viral protease, and that corresponds, if you will, to viral replication, almost in a one-to-one relationship. So I think for the first time, we have at least some insight and potentially insight into COVID patients of why some of them get very severe disease and some seem to have fairly mild disease because of alterations in the interferon response. So one other point about the experimental work before getting into the clinical piece. The other thing that we know about this is it's not only control of viral replication, there's also activation of not only type 1, but also a so-called type 2 immune response in the SARS model. So I showed you earlier, if you look at STAT1 null mice, they get fairly severe disease under these conditions. You can see that here histopathologically again, wild type mice versus STAT1 null mice, quite severe disease. We expected that to be due to interferon signaling and so on, but it turns out also to be due to the development of a type 2 immune response, because if you cross those mice with STAT6 mice that have a defect in controlling this type 2 immune response, I'll explain to that to you in just a second, you can see that they don't develop the progressive disease that we see with interferon deficiency. And as a marker of that type 2 response here in so-called M2 macrophages that are a big part of the type 2 immune response, you can see they're highly activated under native conditions, but essentially gone if you cross the mice to a mouse that can't no longer develop a type 2 immune response. So what does that mean? I think more in simple terms, I don't want to drag you guys into too much cellular immunology here. What it means is, two things. First, there is a type 1 response. It needs to be in balance to make things work well. If you get rid of it, you get into trouble. If you get too much of it, you can actually, in some cases, have a benefit. But in addition to that balance, there's also a balance between type 1 and type 2 arms of the immune system. So this response, which is driven by different cytokines now, such as IL-4 and IL-13, leads to overlapping but different signaling components, can actually lead to progressive disease, at least under experimental conditions. And I hope to show you that that might be a strategy for fixing these things for COVID in people. Okay, so enough prologue. What do we know, how to make some sense out of this for what we're seeing in the current conditions? So I just preface this by saying the literature here, while incredibly abundant, and our Chinese colleagues are very prolific in this contribution, in spite of being highly abundant, is generally suboptimal, to use a polite word. So I picked and pulled here from the various pieces of literature to try to give you some overall picture of what I call data for what's happening in this patient and these patients. And I want to emphasize a couple things. First, this male predominance that I mentioned we see experimentally, and we also see clinically, something to look out for and something I can tell you that we don't understand. The other thing that I want to come back to, which I've tried to introduce experimentally so far, and I'll show you some more of that, is it's not all happening in the first few days of illness. And when I first heard about these reports, it made perfect sense to me based on what I knew. You can see here in this particular study, it's 10 days before they even come to the hospital, recognizing that the peak of viral replication is, is 3 to 5 days most likely after initial infection. So it's a delayed admission and then another delay until they get fixed. So it's really a couple weeks, 2 to 3 weeks really, until the patient progresses to the point of significant mortality and morbidity. The other thing that I just want to highlight here that we've come to recognize is that there's a fairly significant, I don't know if you want to call it, I call it a vasculopathy. Others in heme will probably call it a coagulopathy, but a lot of the parameters for clotting, low platelets, low hemoglobin, high D-dimer, prolonged clotting times, and eventually some degree of acute kidney injury and inflammation fit very well with a picture of microangiopathic coagulopathy that we'll talk a little bit more and leads to certain implications about this disease. I think this likely reflects endothelial cell injury, as I'll explain in a minute. There's not much pathology of any worth so far, but what has been pushed, especially from the Chinese sector, is the fact there's significant alveolitis and alveolar disease that they say represents ARDS. And that picture for ARDS was pushed and continues to be pushed quite strongly in many sectors. But what we've come to learn, both from our European colleagues and from the patients here, lung compliance is essentially normal, even as the disease progresses to some degree. So this is probably not, at least in any classical definition, ARDS, which is characterized almost uniformly by having stiff, not normal compliance lungs. And the other thing that I want to emphasize for you as the disease progresses, both bronchoscopically, if people are brave enough to do that, and just by looking at secretions of these patients, there's significant airway disease in terms of purulent sputum that's reflected under these conditions as well. So it is an alveolar disease, but it's also an airway disease, and as you well know, it's also a nasal disease. So it's a typical top-down infection of respiratory virus. Starts in the nose, moves to the airways, and moves then down into the distal lung spaces to give all of the signs of clinical symptoms that we understand are part of this syndrome. So what we know now, just to crystallize this to some degree, where we stand, first, it doesn't look like ARDS. I think that was a misnomer to some degree. Should be ARDS, looks like ARDS, all that sort of thing, but at least not initially. So the ICU guys will tell you that they've learned that you can oxygenate very conventionally without ARDS measures, and think of this more as a situation for hypoxic vasoconstriction and shunting, which can be corrected without such drastic measures. So good news there. I mentioned already that their second clinical feature of this is this microthrombosis, and in some cases, perhaps macrothrombosis, although that seems to be quite rare. I think this is likely due to the fact that the viral receptor is on endothelial cells, in contrast to some of the other respiratory viruses that causes an endothelial cell damage response that likely leads to this problem. But from a practical standpoint, we've been very vigilant, and others have now been very vigilant about anticoagulation and the need to protect damage from that sort of mechanism. And the third thing, which is where I'm going to spend all the time left, is this issue of progressive disease. So this isn't all happening like a cold sort of syndrome. And that raises the interest in the possibility that there's an abnormal immune response, as I've already mentioned a bit, and the need to define and somehow modulate this response, which we think is altered in a bad way, correct it to a good way, and then prevent some of the morbidity and mortality that's occurring here. So it alters this diagram that I showed you earlier of acute illness, because now we have a second wave of disease. I'm going to call it progressive disease for the purposes of this sort of conditions. And we don't really know the time course. This is the time course that we've mapped out for some of the phenotype in terms of inflammation and tissue remodeling. And this is over here, what we've subsequently learned about what might drive this downstream phenotype, and fits really pretty well with what we see in COVID patients in terms of beginning around a week, 10 days or so after infection, rapidly getting worse through mechanisms that I'll show you, and then continuing on until it gets resolved, hopefully by proper therapy. So a little bit more on COVID itself and what might be underlying this. I mentioned already that there's some interest in trying to figure out what cytokines might be abnormal under these conditions. And the one that people have latched onto firmly, as they have done in other forms of sepsis and other epidemics, is IL-6. And you can see it fits this same time course that I mentioned earlier for what we often see in this kind of paradigm. And that is, it's not until 2 weeks, even 3 weeks or so after onset that you start to see increases in these kinds of levels. So this is everyone's favorite at the moment. We have a trial here. There are many trials across the country and now across the globe for monoclonal antibodies directed against IL-6 or IL-6 receptor, in the hopes that this is functional and not just a marker of the inflammatory response. So is that true? We don't know. It hasn't been true yet, but hoping for the best here. So unfortunately, it's not as simple as IL-6. So this is from a Chinese study that looked at a whole panel of cytokines that get altered during this time after severe disease in COVID patients. Here's IL-6 up here on the right, and you can see that, in fact, in this particular study, it's elevated in only a very small subset of patients and has very little relationship to severity of illness. So whether this works or not, we don't know, but you can see already that I have some skepticism as to that possibility. And in addition to the issues directly related to this, you can see it's not alone. The other type 1 cytokines are also upregulated: interferon, MCP1, TNF, just to mention a few, IL-8. And in addition, not only is there upregulation of the type 1 immune response as we predict, but our friend here, the type 2 immune response in terms of IL-4, IL-5, IL-13, IL-9, and so on, are also upregulated, may or may not be in relationship to severe disease. And just to make things even more fun, the third arm of the immune system, the so-called type 17 response, is also upregulated when patients with COVID get ill. So to put it in a nutshell, there's a lot of things going on, and we don't really know yet in COVID itself which one of those might be responsible for driving this progressive disease. But lucky for you all, and unfortunate for COVID, we've been working on this problem with other viruses for about 20 years now. And I want to show you how we solved it for other viruses for 2 reasons. One, to show you it can be solved, at least to some degree. And secondly, that it might fit with a lot of what we're seeing in patients with severe coronavirus disease. So first, mice, I promised to introduce at least some of the studies in humans here for what causes this progressive lung disease after severe viral infection. And we've used any number of models to test this now. One of our favorites is a natural mouse pathogen. It's a mouse parainfluenza virus known as Sendai virus. As I showed you earlier, very closely related to other RNA viruses that occur in humans as common pathogens. And the interesting thing about that If you use that virus, infect the animals, of course you see viral replication, as I mentioned, that peaks out early on in the course. The virus is subsequently cleared a couple weeks or so after infection, as it should be. But instead of your disease getting better, it actually turns out to ramp up 10 to 21 days after infection and then get even worse later on. So then in spite of the fact that the infection has been cleared by the body, for some reason, it goes on and get this progressive inflammatory disease that obviously leads to significant mortality and morbidity. And as I say, fortunately for us, we've spent a long period of time trying to figure out how this happens. So experimentally in animals, mice, pigs, other models, and validated in humans, I'm going to end up with just the bottom line cartoon here to tell you that these kinds of infections infect primarily epithelial cells, both airway and alveolar. They reprogram these cells, particularly a stem cell subset of that population, to proliferate and expand, presumably as a mechanism for repairing the lung. But then for some reason, in some cases, they see that this is a continuing danger, go on to produce cytokines such as IL-33 that drive a type 2 immune response, that activates type 2 immune cells in the innate immune system. They release cytokines that cause further inflammation and particularly cause inflammatory mucus production, which leads to a lot of the morbidity, mortality, and the phenotype that I showed you is true in these patients, including COVID patients. So this we're pretty certain of, in fact, experimentally. And the question for us now then was, what can we do about it? So as promised by the title of my talk, I want to try to lead you down the road of where we're at with trying to fix this thing. So the first thing that we needed to do there was figure out something that's druggable. And the classic issue there would be some kind of signal transduction pathway, and when within that family, often a kinase. So we wanted to place some kind of druggable kinase in the pathway that I just showed you in order to have a target for a pharmacological approach. And we did that by simply creating cells that can express airway mucus, thinking that that was a significant phenotype for the kind of disease that develops after viral infection. We then ran those cells against a panel of kinase activation markers and tried to determine what, if any, kinase might be involved in this pathway that hadn't been identified previously and might be approachable pharmacological. So we identified then a kinase known as MAPK13. It hadn't, sort of orphan kinase that hadn't been studied under these conditions or others. Most of pharma had concentrated on the MAPK14 pathway, had been unsuccessful there, as you will probably well know. And we thought initially, well, that makes sense because they're targeting this guy, not this guy, and we therefore have a candidate for pursuit. To show you that this also occurs in people, we've looked at a number of different conditions in patients who have inflammatory lung disease and signs of airway inflammation. This particular one is taken from COPD patients who have overproduction of airway mucus and consequent inflammation. And you can see here too, very similar to what we see in cells in mice, there's activation of MAPK13 along with its cousin, MAPK14, which had been targeted previously by other pharmacological approaches. So not surprisingly, we set out then to develop an inhibitor of this particular kinase using a structure-based drug design approach, marching through the system, starting with an old chemical scaffold from Boehringer, shown here, that has at least some weak activity against MAPK13. You can see it's almost in the micromolar level. I might say that we consider that weak. All of the approaches that I mentioned to you earlier that are being applied to COVID are in that range. So another reason not to be too optimistic about some of them. But it was designed as a MAPK14, nanomolar potency, and we wanted to redesign it, if you will, to better hit the target that we thought it caused post-viral disease. We used the structure of this molecule to figure out how to do that by modifying different parts of the compound binding to different parts of the ATP binding pocket. I better move along and not spend too much time there and just cut to the bottom line. We eventually found something that seemed to work. So here, first, just in human airway epithelial cultures, screening some of our top compounds out of our about 500 that we've generated. You can see that some have very good effectiveness against blocking mucus production. And we selected one of those, again now potencies in the very low, this case, picomolar range for effectively blocking mucus production in response to IL-13 stimulation of primary culture cells. So at least in vitro, we thought we had something that worked pretty well in terms of correcting this problem. So just a couple points now that we've been able to take this on now to in vivo studies based on the pharmacological properties of these compounds. So not only does it block the inflammatory sort of phenotype in vitro, it also blocks acute illness after viral infection. We've done this in a mini pig model where we can challenge the pigs bronchoscopically with virus, measure what's going on through consecutive BAL samples with and without treatment. And you can see here on the left, if we give these animals infection, the virus replicates. It causes mucus production, in this case, a fairly short-term illness that subsequently goes away, in addition to inflammation, in this case signified by a type 2 sort of response defined as eosinophils. And if we instead treat these animals now with oral dosing for this compound, you can see it doesn't affect viral clearance, good news, but is able to effectively block inflammation and mucus production. So it blocks the mucus formation, and then most importantly, which is really, I think, what we want to try and pursue in the case of things like COVID, it also blocks the progressive lung disease that we see in the mouse model. So in this case, same sort of thing, inoculating the mouse with a respiratory virus, following airway inflammation, mucus production, lung remodeling, and so on, with and without treatment, now going out to 7 or so weeks after the initial infection. You can see the acute illness, not much changed by treatment in comparison to vehicle. But if you look at all of these parameters that might be important under these conditions, mucus production, mucus cells, type 2 cytokine production, the alternative macrophage activation that I mentioned early on in the talk, all of those things can be attenuated significantly by treatment, starting not only before infection, but also attenuated significantly even when we start treatment at around 10 days after infection, which is obviously more realistic in terms of clinical terms. And so it not only corrects this problem in terms of inflammation and mucus production, it also corrects this basal cell stem cell activation and hyperplasia that we think drives a lot of this disease. And so here the same sort of protocol, now looking at basal cell markers here, aquaporin, keratin 5, P63. You can see there's significant attenuation by treatment, even when given after the basal cell hyperplasia starts. And as a result, if you compare the lungs of animals that are untreated compared to those that are treated, you can see even at this low power that all of the mess that occurs in the airways and the parenchyma of these animals as long as 7 weeks out from infection are very significantly attenuated if the animals are treated with this drug. So what does that mean? Just quickly trying to put this again in a cartoon that I hope helps you think about what's happening in this disease and gives you some confidence that at least one guy can do something about it, or one team, I should say, can do something about it. First, there's this issue of mucus production, which is a significant problem in real life, of course, but and in some cases is a cause of significant morbidity and mortality. And here, this kinase plays a significant role in this process, and inhibiting it can attenuate. But in addition, I want to stress this concept that something like cancer is occurring, and that there's also activation of a stem cell population under these conditions, also dependent on MAPK13 and related cell cycle dependent kinases. That's also inhibitable by a similar approach and can downregulate this response and thereby attenuate this progressive disease that we see, I believe, in the ICU that drives the morbidity and mortality here. I won't have time to tell you that we validated this in human cells, also in stem cell populations, also in organoids, and even in related cancer cells where this same kind of mechanism seems to get activated. So, pretty close to being on time. So what have I tried to tell you? I think the bottom line is depicted here, two major takeaways. I think if I can stress that. The first, defining the basis for the acute entry. So that's how we started this talk. Those are the people that are doing it, and it really depends on the art of pulmonary and critical care medicine, particularly in terms of respiratory and other organ support. In this particular case, also some effort in terms of anticoagulation and in terms of protection and treatment of the resultant end organ damage, particularly in the heart and the kidneys, and the additional eventual application of antiviral drugs in the form of antibodies, passive or man-made, and eventually perhaps an antiviral drug. As you know, I feel somewhat skeptical about the current approaches, and even more skeptical that those might apply to the next and future kinds of viruses that might cause this problem. And that gets us to defining the basis for this progressive disease that's found here and really in virtually every respiratory virus that we've studied to date. And that depends on then, instead of pulmonary and critical care, my other hat, the art of viral immunology. And I've tried to just give you one example of a smart, we think, and rational scientific approach to this issue. First, defining the immune response, what kind of druggable elements are in that response, and then developing some sort of inhibitor in terms of a drug that can be applied to prevent it in the future, treat it potentially in the present, and correct it in the long term. And then lastly, I just want to get back to my initial elephant story here. So we've got the elephant cornered here, we think, in terms of our drug discovery efforts in Washington University. And these are the people who I need to thank for doing all of the work that I showed you, both in terms of the acute response, people still in the gang here, Young and Dailing, the chronic response, particularly Canyon, Ying, and others who have been or left the lab, the epithelial response covered by these guys, the preclinical models, which are desperately needed under these conditions. I showed you mouse and pig, the extension to humans, and the ability to make and develop drugs in the pharma environment of Washington University. So I'll stop there. This is for you guys to get credit for putting up with me. And then I'm happy to take any questions if you have them. Thanks.
Dr. Holtzman,
Yes.
do you see the Q&A at the bottom of your screen?
I don't see anything but I still am on my, should I escape?
I'm sorry.
Should I hit escape?
Yes, you can.
Maybe stop share. Should I stop share? Oh, here's it.
I can take your questions. I can ask them if people want to text in.
Okay.
Here's one question. What's his best guess for the male-female?
So Dr. Holtzman, a question from Dr. Frazier, and the question being, what's your best guess for the difference between male and female?
You know, it's a relatively understudied subject, so I have to, full disclosure here, we've used male mice almost exclusively for the last 25 years, really just in an effort to minimize differences in the laboratory environment. There is some work looking at hormonal influences, you know, in terms of estrogen, progesterones, and so on. And there is some evidence that they might influence the immune response under these conditions, but we really don't understand the difference. Similarly, we have a hard time understanding the effect of age and circadian rhythm and biological clock and all of those things as well. So gender and age influences are not well defined. And in fact, you know, what I showed you, I get some bragging rights here for the group. Even even the whole issue of understanding what happens, you know, pathogenically after this type of condition, what I showed you is kind of what there is in terms of understanding this response. So if even just this initial definition needed to be done before we can now drill down on determining what kind of hormonal and age effects there are on the components that I showed to you guys.
Anyone else? Welcome to chat. I'll relay any questions that you have.
So Mike, another big difference we see is tremendous impact on the African American community with worse disease. And, you know, it's been speculated that's related to access to care, underlying comorbidities. Do you have any comments on racial differences and health disparities and outcomes?
So yeah, so I like you say, I think a large part of that is based around, you know, delivery of care and optimal care in that community. But, you know, with the same kind of condition, I'll just put on the asthma hammer for a second here. If you look at where morbidity and mortality occurs in that disease, it's also reflected. So we've lived with this issue in the airway disease world for a long time and tried to understand, at least to some degree, why that's occurring. As you know, that's also a disease that's largely driven by a type 2 immune response. And they're also linked, you know, to increased morbidity and mortality by sex and by race and by age. So the likelihood is that some of the genetic susceptibility is linked to race. I tried to give you really just our very first example, the old DTX3L SNP here, way back buried in the talk. Really just our first example of why we think there might be genetic susceptibility to this type of disease. So in the long run, perhaps, we can start to link some of those SNPs into the human population and then determine whether they are or aren't connected with things like race, age, and gender. And I would be surprised if, as we expand our understanding of that genetics, if that doesn't come up into the story. That particular, you know, gene that I mentioned is a type 1, obviously immune response gene, since it's involved in the interferon signaling system. But there are other genetic linkages in type 2 immune response genes. None of that has been defined in a significant way beyond just the first cut. But understanding those, I think, will let us understand why certain subpopulations of people are more susceptible to disease and more susceptible to this progressive disease that I think drives a lot of the problem.
Thank you.
Okay. Question from the audience. It's that the preclinical data for the small molecule kinase inhibitor to block post-viral inflammation seems quite promising. Question is, what's the status of moving this inhibitor towards a phase 1 trial?
Good question. So we're currently doing our final safety studies. I wish we were ready for COVID. It pains me that we're not ready to give it to patients, but particularly when I see what is being given. So we're in the final preclinical safety testing. The FDA hasn't cut us any particular breaks. It all looks fine for oral dosing. I'd expect to be, you know, all things being equal, I'd expect to be into safety studies in humans by the fall, perhaps, if everything goes well. Not surprisingly, we were delayed, as everyone else is delayed, by the shutdown. So a lot of our preclinical safety studies are being done, ironically, in China, mostly in Beijing, not Wuhan, fortunately. And that group is now coming slowly back on board with their capabilities for continuing the testing. So we've been set back, just like everyone else has, by the epidemic. And we're set back now a little bit in terms of just getting materials from China to the US in terms of samples that we'd like to analyze. So we've been delayed, but in spite of the delay, if everything goes okay, we hope to be into human safety studies in the coming year, and then be ready, if you will, for the next respiratory virus when it comes a year or two from now.
Okay, another question from the chat is that, do you have any thoughts about the BCG vaccine being protective against COVID?
So great question. With all these things that I forgot to mention. So BCG is very interesting to me because when I saw that, I thought it fit just perfectly with the things I've been trying to tell you. So BCG is, somewhat, in sort of rough terms, of a type 1 sort of / interferon kind of stimulator. So one could imagine if one took an agent that skewed things towards a type 1 interferon sort of system and away from a type 2 sort of cytokine system, it would fit very closely with the kind of paradigm that I tried to lay out for you guys. So it doesn't surprise me. In fact, it might even be predictable. Kind of gets back to the argument I gave you up on the top of the argument with the SMICE compounds. So compounds that might drive interferon signaling response in a productive rather than harmful way might skew the immune landscape, if you will, towards type 1 and away from type 2, might be a benefit under the very sort of paradigm that I tried to lay out for you. So it doesn't surprise me in immunological terms that that's occurring. Now, is it effective enough to handle what we're seeing in the intensive care unit? I'd be surprised if that were the case. Does it give you some advantage? That wouldn't surprise me at all.
All right, in the interest of time, this will be our last question. So we'll finish with a two-parter for you. With it, the question being, we have heard that corticosteroids have a detrimental effect, but given the overall activation of the immune system in progressive immune-mediated disease, how do you possibly explain that? And part two of that question will be, could they be more effective if administered during the progressive stage?
So the steroid hammer. So not surprisingly, it's been tried in every other disease, including this one, and it will continue to be tried. I think the initial concerns, I think this is the first part of the question, the initial concerns is the immune compromise that steroids convey, and in any setting of infection that can be problematic. So I think a lot of our concerns about both primary worsening of infection and secondary infection in this setting using steroids were a concern. And for that reason, while it was tried, I don't think it showed any clear benefit in the helter-skelter fashion that it was used. And because of the concerns for infection, most people backed off. Now, as you can probably predict, that seesaw is tilting also to where people are seeing these cytokine responses and inflammatory effects and trying to figure out if there is still some benefit under these conditions. It's been thrown out for ARDS, I believe, effectively a long time ago, and I don't have any significant faith that it will be of much benefit here. I will tell you, in the model that we've studied here, the one I presented to you, which we've studied over the years, there is some effect, beneficial effect of corticosteroids, and that's because they dampen down the alternative macrophage response that I mentioned was part of this syndrome. So from that standpoint, they do have some benefit, but it's not nearly the kind of benefit that I showed you for when one truly understands the immune response that's occurring and tries to specifically target that kind of response. That, to me, is a much more rational and effective approach than the sort of the spaghetti on the wall thing that you're getting with a corticosteroid effect. Having said that, people are and will continue to try to figure out some treatment window where the steroid effect happens to be beneficial. I don't think those are done in any truly controlled fashion, and I would argue that they're almost never done with the kind of immune profiling that I think you would need to understand if they're working and why they're working and how to improve what they're doing.
All right, thank you so much, Dr. Holtzman. If I could ask you real quick, do you mind just putting back up the QR codes just so other people can sign in real quick? But otherwise, thank you so much again, and we look forward to continuing this next week with our next talk.
Thank you all.
Thanks, Mike.
Sure.